Well, thanks so much for coming out to see this presentation today. I'm going to be talking a lot about blueberries and a few different things. Before we get started, though, I kind of wanted to talk about who I am. My name is Dr. Kendall Lee, and I am a USDA postdoctoral fellow at Hudson Alpha Institute for Biotechnology. But I am also a co-founder of a company called Vail Genomics. And so I'm in a unique position kind of spanning both these worlds. And the work I'm showing today reflects that intersection. All of the data was actually generated at Hudson Alpha, but a lot of it is like what we also do at Vail. I also realized I'm wearing the exact same outfit that I was wearing in this like headshot, so maybe I need to like go to the mall after this and over there and expand my business casual. So this is my blueberry buy-in slide. I don't think anyone really needs to be pitched on blueberries, they're awesome. But in case you don't, this is my pitch. Blueberries are an incredible crop. They're not only delicious, but also nutritionally powerful. They're packed with anthocyanins, fiber, vitamins. If you can eat them every day, you definitely should. But they're also economically important with about $3.7 billion market value last year. So they're kind of that perfect crop where they're good for you, they're good for the economy, and they're tasty. All right, so today we're gonna talk about a lot of stuff. First, just kind of get into blueberries. Then we're gonna talk about the Pangenome I built and then long-read low-pass mapping to that. And then we'll end with just a little bit of sci-fi. Okay, so before I begin, I just want to thank Dr. Juliette Chu. She is the University of Georgia blueberry breeder, and she's the blueberry expert, so none of this would have happened without her. One of the really fascinating things about blueberries, or one of my favorite facts, is that they're one of the only crops that are actually native to the United States. So I think that's pretty cool. And here you can see the map shows main blueberry production regions, and it really spans the whole United States. So that means we have different types of blueberries and different species of blueberries that we grow. So up north, where it's colder, we have northern highbush. And in the southeast, we grow southern highbush and rabbiteye. And they differ based on chilling hour requirements. And since I am working with a University of Georgia breeder, we are focused on these southern types of blueberries. So southern highbush is the main one that I'll be talking about through this presentation, and it's the most popular. Vaccinium corymbosum, it is an autotetraploid, and it is an interspecific hybrid that was bred specifically to be grown in the southeast. There's also rabbiteye, which is more of an enigma. It's a different species. It's a hexaploid, and it's native to Georgia. We didn't breed it, it was already there, and less is known about it, and I'll get to that at the end. So in the scope of my postdoctoral fellowship, we have a few traits that we're looking for, trying to uncover the genetic basis of these phenological traits. Flowering time, fruiting time, and the time between flowering and fruiting. And also fruit weight, because we all like big blueberries, right? And why is this important? So the timing of bloom for blueberries is critical because once you have that flower, if there's a late season freeze or something, that will wipe out, that'll kill the flower and then wipe out production for the year. And that actually happens quite often in the southeast, and it's devastating to farmers. Similarly, with the time between flowering and fruit set, in the southeast, I'm from Georgia and I've been living in Alabama. It is so hot and humid, and we all know that. So the longer it takes for that blueberry to develop, the more risk there is of issues like fruit softening, skin splitting, fungal rot. And so we would like to be able to shorten that time so we don't have this effect on fruit quality and yield. And we all are trying to do molecular breeding at this point. We know how important it is. I just want to make the point that it's particularly important in blueberry. Blueberries are a perennial species, and if we make a cross and have a new seed and plant it in the ground, it'll be years until we could even see a berry to evaluate. So being able to have markers to speed up this production or the selection cycle is really vital in blueberry breeding. All right, moving on. So for this work, I'm working to develop resources specifically for University of Georgia, and the first thing we needed to do was make a southern highbush reference genome. And so we chose Suziblue, who I have a love-hate relationship with her. It's been difficult in the wet lab, great on the analytics or on the genome assembly part. But it's a southern highbush blueberry cultivar that's popular out of UGA, and it's a founder for a lot of the population that we're studying. And I was able to assemble this genome, of course, with HiFi and Omni-C. I have to shout out Zach Mahark from the Harkis lab at Hudson Alpha, who really kind of showed me the ways of manual curation and juice box, which we had to do a lot of. And this is a contig plot from GeneSpace just showing the contiguousness of all 48 chromosomes, and the telomeres are where they're supposed to be. I'm going to move on quickly through this genome stuff since we have a lot to cover. But once we had the southern highbush reference genome, we were then able to sequence 5 other southern highbush genomes and get them to the contig level assembly, and then do reference-based scaffolding. And I was able to do this recently thanks to another Harkis lab member, Laramie Kozbek, who made a tool called, yet another reference-based scaffolding tool, YARBS. And what's so good about this tool is that when I feed in my blueberries into like ragtag or some of these other scaffolders, because it's an auto polyploid, the scaffolder makes some wonky decisions. And this tool allows you to manually make changes yourself and go in and actually curate it. So we go from something like that to something like this. And quickly, this is just a synteny plot between the 6 primary haplotypes of these 6 genomes. And there's some collapse missing a little bit, but overall, I'm pretty happy with it. And then I built the Pangenome using KhufuPAN, and this is an open source pipeline. It's a wrapper for building your Pangenome, mapping to it, and then calling genotypes. And so this was made by Dr. Walid Kourani. He was in the academic lab at Hudson Alpha, and now he is an employee at Vail Genomics. And so, of course, we got to talk a little bit about the Pangenome because it's always fun to start looking at the diversity there. So what you're seeing here is out of PanCamer, which just camerizes the genomes and starts to look at the diversity. And if you look at the collection curve, that top line, that's that Pan curve, every new genome we've added is continuing to go up, meaning like every new genome is adding diversity. So this just kind of speaks to the need for a Pangenome in a species like blueberry. Okay, now long-read low-pass. So Jake already kind of introduced this topic, but just to go over it again with an example, if you have a 1 Revio SMRT Cell, you can get, I'm using round numbers here, we can get more than 100 gigs off a Revio SMRT Cell now, but I need easy math. So if you could say 100 gigs, if I put 1 blueberry, autotetraploid blueberry on there, I could get 41x coverage and get a genome assembly. And that's great, and that's how we've typically used HiFi thus far. But we wanted to further expand that and actually use long-read sequencing for genotyping. So we'll take 12 samples of blueberries, put them on 1 Revio cell, get more like 3.5x coverage, and then map that to a reference genome or a Pangenome and get really high-quality genotyping data. And so that is what we did. We kind of did this like segregation analysis type experiment where we took the blueberry selections that had extreme ends of phenotypes, so the high bulk and the low bulk, for fruit weight, flowering time, fruiting time, and we sequenced them with both long read low pass and then also with short read, so we can do a comparison, and targeting about 1x step for both of them. And we found that long read low pass, just kind of blew the short read out of the water in terms of more variance. 32% more variance overall, a ton more structural variance, and I can talk more to you about that if you're interested. And I'd also like to point out that if we were using just a single reference genome, the short reads would not have found any structural variance at all. The only reason they were able to call any is because it's a Pangenome. And a lot of that is due to the whole genome coverage. Jake kind of showed that earlier. It's not as dramatic in blueberry as it was in peanut for whatever reason. I think we just got really good short read data, but we still had significantly better whole genome coverage with the blueberry than we do with the, with the long read low pass than we did with the short read. And what does that actually get us though? Because the whole point is to actually do the breeding work. So as part of KhufuPAN, we have something called QTL-VAR, which just analyzes the genome for where the genotype is differing between those high bulk versus low bulk, to give us a clue where should we actually look for these candidate SNPs, candidate markers, because this is where they're very much different in the genome. And that's what this peak right here is showing us. And you can see the peak on long read low pass. The short read low pass data totally missed it. So like I always talk about, when we're doing this kind of genotyping work, it's like trying to find a needle in a haystack. And if you don't have half the haystack like you do with a lot of short read sequencing, you might miss it. And these are just some candidate SNPs I pulled out. I just pulled out a few. I found a ton. But it would be such a shame to miss these types of SNPs where we're getting 30, 35% variant explained, highly significant allele effect. And that is what long read low pass is able to get us. Cool. Now we're going to switch gears totally and talk about CiFi. So Jake already introduced CiFi, but just to reiterate, CiFi is basically just long read based chromatin capture. So it's like doing Hi-C, but with long read. And Jake knew that I had been really struggling with some of my blueberry work. And so he called me and said, do you want to try to make this work for your blueberries and do some R&D? And I said yes. And thanks so much to the Dennis Lab and especially to Sean for helping, emailing with me and talking to me, especially when it came time to do that analysis, which I'll talk about in a second. And this is where we're going back to our rabbiteye genome, because it has been so difficult to try and get something going for this guy. It's a hexaploid, but not much is really known about it. There's pretty much no genomic resources available. So and everyone's really excited to do some comparative genomics and see what's actually going on there. So I really wanted to get this genome done. So what we did is we took CiFi and adapted it to Omni-C, because Omni-C is what has historically worked best for my blueberries. And I spent a lot of time with different methods and trying to get this data for blueberries. So I'm going to kind of skip over the details here and how we did this for the sake of time, but just know it mostly has to do with decreasing the fragmentation step, and then at the end we're doing, instead of the normal PCR that is included in Omni-C, we're doing PacBio amplify, and then we are prepping it to go on the Revio instead of a short read sequencer. And from that, we were able to do an exact comparison. I had one cultivar from UGA called B080, and we had all the CiFi data, and we fed it through HiFi-ASM. And so we had both standard Omni-C for this, and then we also had our new CiFi-adapted Omni-C. So we were able to actually really compare this. And then once we had the data, we fed it through kind of your normal scaffolding pipelines with Yas and Juicer. And both took some manual curation, but I spent a really, really long time with the standard pipeline before I even got to doing CiFi, because we thought, was going to be kind of our one shot at this. And even with 41x data, with that Omni-C data, you can see these chromosomes are pretty truncated. And this is B080, what I call the primary haplotype. I basically pulled out the most complete chromosomes I could find and made it one haplotype aligned to the Suziblue reference genome. And so you can see that they're pretty truncated. They're not capturing the full span of it. But then with only 4x of data, of coverage for CiFi and an average read length of 1.5 kb, I was able to manually curate out very complete chromosomes from each chromosome block. So we have 1 through 12 represented. So now this is our jumping off point to actually start doing some work with rabbiteye, which is very exciting. And just like a sneak peek for that is another gene space plot where we have B080 and W85, which is a diploid blueberry reference genome, and Suziblue. And you can see they're all highly synthetic. So this is our first kind of intro to what is going on with the rabbiteye genome. Okay, so my takeaways. Blueberries are awesome. I hope you guys got that from this talk. But there's also there's traits of interest that are really important in the Southeast particularly that we need to be addressing, and that's what we're working on, building resources to do so. Long read low pass identifies key variants that the short read just misses. The structural variation And I didn't mention this earlier, but we also filter out a lot less data with the long read. So it's essential for finding that needle in the haystack. And then CiFi. CiFi can be adapted to plants to help resolve complex genome assembly. And we really just kind of were getting started with that. So that was like our first pass, and we already got a whole rabbiteye primary assembly. So with that, I would thank you for your attention. Thank you to the Clevenger Lab, the Co-Lab, my mentor, Josh Clevenger, Alex Arcus, the other co-founder of Vail. And please check out Vail Genomics booth 403. And I'll be around after to answer questions.